Hello, my name is Maddie Kirklin and I am a 9th grader at Live Oak Classical School. This is my 3rd year participating in science fair. My 7th grade project focused on which types of aquatic plants are best at filtering water with respect to E. coli. I determined that Equisetum himalaya was the best variety of the plants tested. My 8th grade project focused on the minimal amount of Equisetum himalaya that would still filter E. coli effectively. This year, my project idea was to determine if there was a structural component that contributed to the plant's ability to filter E. coli from water. However, after further research, this concept evolved into the question as to whether the natural flora living on the surface of the plants contributed to its ability to filter water effectively. It was determined from last year's project that samples from the water where Equisetum himalaya was grown showed a reduction in E. coli concentration when compared to samples from water where typha was grown. Therefore, the question could then be asked, is there a specific bacteria that grows as natural flora on the surface of Equisetum himalaya stems that serves to reduce E. coli in water supplies? My hypothesis was that if Equisetum himalaya and typha are swabbed and bacteria is identified, there will be a bacteria found as a natural flora of Equisetum himalaya that serves to control E. coli growth. To begin the experiment, 5 plants of each species were established for 2 weeks. After they were established, 3 stems of each plant were swabbed and the bacteria was transferred to nutrient auger plates. After 24 hours, colonies were inspected for the form, elevation, margin, color, and diameter of the bacteria. Then the gram staining technique was used to identify if the bacteria was gram-positive or gram-negative, and the bacterial shape and arrangement. I would have tested the water for E. coli, but the Mount Carmel water treatment plant was closed due to COVID. However, 2 years ago, my project showed that Equisetum himalaya was more effective at filtering E. coli than typha. Both Equisetum himalaya and typha have natural bacterial flora that live on their stems. The result of this experiment shows the following bacteria and their representative percentages as found on Equisetum and typha, respectively. Gram-positive Streptococcus, 15.7% and 13.3%. Gram-positive Streptobacillus, 11.8% and 16.7%. Gram-positive Monococcus, 9.8% and 16.7%. And gram-negative Streptococcus, 11.8% and 13.3%. These 4 types, as identified by gram stain and colony formation, made up a significant percentage of bacteria found on both plant varietals. Interestingly, gram-positive Staphylococcus were found only on Equisetum himalaya. Therefore, my hypothesis seems to have been correct. This project applies to the present day because E. coli is a dangerous and all too common pollutant in our freshwater supply. If we can determine what makes Equisetum himalaya filter E. coli out of water, it might be useful in medicine or as a means to provide safe drinking water for our planet. I really enjoyed looking at the bacteria slides under the microscope and getting to work with plants again this year. If I continue this project in the future, I would try to find the names of the natural flora present and see how they kill off the E. coli. Thank you.